Great. Well, welcome everybody, and thanks so much for joining us to have an introduction to participating in the Pacific Northwest Bumblebee Atlas in 2025. We can just start with some intros. I'm Molly Martin, and I'm an endangered species conservation biologist with the Xerces Society. And I'm based in Portland and co-lead the Pacific Northwest Bumblebee Atlas with Rich. Do you want to introduce yourself, Rich?
Sure. Yeah, my name is Rich Hatfield. I'm a senior conservation biologist for Xerces. I'm also based in Portland. I'm the Bumblebee program lead for our Bumblebee program nationwide, and I coordinate all of our Atlas programs across 20 states. And I'm lucky to co-lead this program with Molly. Really excited to be with you tonight, and thanks so much for being here.
Great, thanks, Rich. So tonight we're really excited to have you all here to teach you about the Bumblebee Atlas program and give you information and resources that you're going to need to participate this year. And this webinar is for everybody. So if you're a longtime volunteer, this will be a really good refresher and some information that's different about 2025. And if you're brand new and haven't heard about the Atlas, this is a great way to kind of get the deep dive into what you'll need to know to participate. The webinar is about 2 hours. We have about an hour and a half of content, and then we'll have time for questions at the end. And this will also be recorded, so if you need to hop off or something, you can watch the recording later. If you have questions during the webinar, you can use the Q&A feature, so it should be at the bottom of your screen. And Rich and I will probably try to answer some questions as we go, but we'll pull some of those up at the end and talk about them as a group. So the Pacific Northwest Bumblebee Atlas is a project of the Xerces Society. I'm guessing many of you are familiar, but Xerces was founded more than 40 years ago and was named for the now extinct Xerces blue butterfly. Our mission is to protect wildlife through the conservation of invertebrates and their habitat. And we now have about 100 staff members spread across the country, and they work in a range of different programs, including native pollinator work, endangered species. We have people focused on aquatic invertebrates, butterfly conservation, pesticide protection, and a whole range of other great work. We're really thankful to Washington Department of Fish and Wildlife for contributing funding for the 2025 year of this program. And then we're also grateful to past funders of the program, including U.S. Fish and Wildlife Service, Office of Conservation Investment, and also the BLM. And then also Idaho Department of Fish and Game has been a really valuable partner throughout the history of this program. Xerces is a member-supported organization, which means that our work depends on the generosity of people donating who are really dedicated to making a difference in the natural world. So a huge thank you if you're already a member. And if you're not yet a member, we encourage you to head over to xerces.org/donate to learn more. And we're relying on donations and membership more than ever right now, so it would be great if you want to take a look at that. Xerces is also fortunate to receive funding from a range of agencies, organizations, businesses, foundations, and individuals. So I just wanted to show a list. It's really heartening to see all these groups that are supporting conservation. And so we want to say a quick thank you. The last thank you before we get started on the content is I want to thank all of our past project participants. We kind of provide the framework and guidance for the Atlas, but it's our wonderful participants who are out there in the fields collecting data and making the conservation impacts possible. So big thank you if you've been involved in the project in the past. So what is the Pacific Northwest Bumblebee Atlas? The Atlas is a collaborative effort to track and to conserve bumblebees across Washington, Oregon, and Idaho. And the Pacific Northwest Atlas began in 2018, and the goal was to increase our understanding of where different bumblebee species are found, and then to track their population trends over time. And the Atlas, since 2018, has spread across a lot of the country. So on this map, states that are shown in green are states that have Atlas programs run by Xerces. So it's really great to see it spreading across the country. So during the initial, there were kind of two phases of the Bumblebee Atlas. During the initial phase, which ran from 2018 to 2023, we recruited project participants from across the 3-state area, and we asked them to conduct Bumblebee surveys. So we divided the states into equal-sized grid cells that were about 50 square kilometers each. And our aim during that portion of the project was to get at least one survey, and ideally more, completed in each grid cell in those states. And completing a survey involves selecting a 1-hectare site and then recording habitat data and documenting bumblebees present at the site. And the bumblebee surveys, for those not familiar, are non-lethal. So that means we catch the bees, we cool them off so that it slows their movement using ice, and then we take photos of them and release them. And we're able to identify those bees based on the photos. So we'll provide more information on the protocol in one of our subsequent sections of this training, so you can learn more about that. So just some quick statistics from 2018 to 2023, over 1,500 people participated in the project. They completed over 18,000 surveys and documented about 42,000, a little over 42,000 observations of 25 species. So that's a huge amount of data, and you can see on the map here the dots showing all of those different bumblebee sightings. This effort has really contributed enormously to the conservation of these species. And we don't have time today to go in depth about the conservation impact, but you can learn more by watching our recent recorded webinar, if you haven't already, on ecology and conservation of bumblebees. And we go into a little more depth than that about what we've learned from the Atlas projects and how that's turned into conservation outcomes. So in 2024, we moved on to phase 2 of the Bumblebee Atlas, and this phase is long-term monitoring. So the objectives of this phase of the project are to continue to learn about how bumblebees are doing and the kinds of habitats and management activities that are most important to them, to better understand species-specific detection probability. So how many times do we need to survey in order to actually find these species? To understand factors influencing occupancy of species, so what's making a species be found somewhere or not. And we're looking especially at some of our imperiled species, so occidentalis, morrisoni, and polyginosis are kind of our main focal species. And then also to track occupancy of these species over time to see how the population numbers are changing and how what that's reflecting about these species' status. The two species we're focused most closely on for this phase 2 are the Morrison's and Western bumblebees. And these are both designated as species of greatest conservation need by the International Union for the Conservation of Nature. Here's some nice photos that Rich took of those two species. So based on where bumblebee species, including the Western and Morrison's bumblebee, have been found in the past, for phase 2, we selected 75 sentinel sites with 25 in each state, so 25 Oregon, Washington, Idaho. And this map shows those 75 sites with the different colors indicating sites surveyed at least once in 2024 and then sites not surveyed in 2024. So the surveyed ones you're seeing in red, the majority of sites were surveyed, and then the sites not surveyed are kind of the yellow and orange, which luckily there weren't a lot, but there are some across all 3 states. Sites circled blue on this map were moved after the 2024 season, and that's mainly because of access issues, public land, and also just challenges getting to those sites. So 3 of the sites were moved only a short distance. And then there's one site in Washington, the one with an arrow, that was relocated to an area just west of Spokane, where Morrison's bumblebee was tentatively found last year. So this year, we're asking participants to adopt one or more of these sentinel sites and then survey at that same location multiple times throughout the summer. And ideally, they would survey 4 times, once in each of the time windows shown at the bottom of the slide. So June 1st to June 23rd, June 24th to July 14th, July 15th, August 4th, and then August 5th to August 25th. And while it would be most helpful for people to survey all 4 times, it's still really helpful even if you can only survey once. So as many times as you can survey is great, but don't be scared off if you can only go to a site a single time. And you may also be able to coordinate with other people who could fill in the other time windows at that same site. And one other thing, so while the Sentinel sites are the focus of the second phase, participants are still welcome to survey at any location outside of the Sentinel sites that they choose. So if you have a site you visited during phase 1 that you really like and want to keep surveying, it's still very helpful and useful for us to have data. So feel free to continue doing that. Some quick numbers from year 1 of our long-term monitoring. We had about 250 people participate. They completed over 600 surveys. During those surveys, over 6,000 bumblebees were recorded and 21 different species. So what's involved in participating in 2025? There are 4 main steps involved in participating in the project. And these are all similar to the steps from phase 1. So step 1 is to select and adopt a survey site. Step 2 is to review the survey protocol and gather materials. Step 3, go out and complete your survey. And then step 4 is to enter your data online. All right, so I'm going to click on this tutorial, which is going to take us through how to select and adopt a site, and it will give you a little tour of how to actually do this on the website. Welcome, and thank you for joining me for an overview of selecting and adopting a survey site for the Pacific Northwest Bumblebee Atlas. This is a space where everyone is welcome, whether you're a longtime volunteer with the Atlas or just learning about the program for the first time. I'm glad that you're taking the time to be here, and I hope that you find the content interesting and helpful. This is the second segment in a 5-part training series. This segment provides an overview of selecting and adopting a survey site. Visiting the Resources tab on the Bumblebee Atlas website to view the introduction segment, as well as the subsequent segments in which you'll learn about the survey protocol and gathering materials, how to complete a survey, as well as entering your data online. As I shared in the first training video, there are 75 Sentinel sites with 25 in each of the 3 states. We're asking participants to adopt 1 or more of these Sentinel sites and survey at the same location multiple times throughout the summer, ideally once in each of the time windows, which are June 1st to June 23rd, June 24th to July 14th, July 15th to August 4th, and then August 5th to August 25th. It's still helpful to have participants survey, even if they can't survey during all time windows. And while the Sentinel sites are the focus of the second phase, participants are also still welcome to survey at any location outside of the survey sites that they choose. When selecting a site, you'll want to consider the access to the site, including travel distance, and whether the site is near a road or requires significant hiking to get into. We hope that participants who adopted a site last year will choose to return to that site this year. In 2024, there were 17 sites that weren't surveyed. You can see those sites on this map. The one likely change to this site location from 2024 to this year is that we may move the site in the Skagit Valley of northwestern Washington to an area west of Spokane, where a Morrison's bumblebee was found in 2024. We're especially interested in finding participants to survey in these sites that haven't been surveyed yet. There's some especially interesting sites that I want to point out, like this one in north central Washington. This is a site where the species Kirby-Ellis has been found in the past. Kirby-Ellis is a rare species that we would really like to track. The site is challenging to access. It involves driving on a Forest Service road before hiking about 10 miles into. So if you're looking for a summer adventure in a beautiful place, adopting this site would be a great option. Given its accessibility challenges, we're looking for participants to visit even if they can only go in a single time. The Bumblebee Atlas website has an interactive map that you can use to explore the Sentinel sites. You'll also be able to sign up for the site or sites that you'd like to adopt on the website. So I'm going to go ahead and switch over to the live website to show you how you can explore the map and adopt a site. So here we have the Bumblebee Atlas website. You can find it by going to bumblebeeatlas.org. You'll start by going up to this header bar, scroll over to get involved, and then say adopt a grid cell. Zoom in to make this a little easier to see. So that will take you to this adopt a grid cell page. You can then scroll down to this interactive map. So we'll say we agree, proceed. That takes us here. We can select our region over here on the left. So I'm going to say Pacific Northwest. That will zoom us in here. That's a little too far zoomed in. So here we can see Oregon, Washington, and Idaho from a distance. And then if you continue to zoom in, you can see the Sentinel sites in these purple and pink. And right now, this is showing the sites that were and weren't adopted in 2024. So you can more or less ignore the difference in colors. Right now, the ones that are just solid purple were not adopted, and the ones that are kind of more pink with a different outline were adopted. So we're going to scroll around and think about what site we might want to adopt. Let's take a look. Oregon 6 is right over Mount Hood near Portland. I'll go ahead and zoom in, and you can see this site here. If you keep zooming, we'll eventually have the color disappear. And now you can see these hexagon shapes that make up that overall polygon. And we'd like people to focus, if possible, on a central hexagon here. And then if they can't find good habitat in that area, they could try one of these peripheral hexagons. So you can see on Mount Hood, the central hexagon actually covers the ski area. So we can zoom in and get a pretty good overview here of the satellite imagery. And then you can see there are these yellow dots. And those are locations where surveys have been completed in the past. So you can see someone named this Mount Hood Meadows. That's the name of the ski area. A survey date. And they found 6 different bumblebees there. So this can give you a good sense of where people surveyed in the past, which might be helpful for thinking through where you could survey now, what might have good habitat. You can also, as you scroll around in the center, it will tell you the ID of that area. So this one's Oregon 6, OR_6. Another layer you can see on this map is BLM land. So BLM land is indicated by these yellow polygons, which there's really not any on Mount Hood. But if we zoom around, there's some areas that have quite a bit, and it can be helpful to see. So let's go check out the Steens Mountains down in southern Oregon, southeastern Oregon, really beautiful area. If you haven't visited, I'd recommend going down there. This is a site that hasn't been surveyed yet, Oregon 19. You can see a lot of this overlaps with BLM land, easy places surveyed. There have been surveys conducted along here. And it's not on this map, but I know that Morrison's has actually been found in this area before. So it's a really neat spot. All right, let's say we decide that we're going to survey Oregon 19 as our site. So we can now go down here and enter our information. So we'll say first name, last name, do our home zip code, Bumblebee Watch username. So you should have your username here. If you need to find it, you can click this tab. This will take you to the Bumblebee Watch website. If you click up here, you can log in. You can go to my profile if you don't know your username. And I can see my username here. Entered my email. I'm adopting in Oregon. So it's going to then ask me Pacific Northwest monitoring site. So that was Oregon 19 that we picked. So it's right here. Then it's going to ask me which time periods I agree to survey in. So there are those 4 different ones. And we'll say I have lots of time this summer and I want to go on a trip down to Southeastern Oregon. So I'm going to go ahead and adopt all of those time periods. If you can't do one, we would just say no. And if you say no, then we ask if you give us permission to share your contact information with other people. We'd like to connect people who have agreed to survey the same site, but not during the same time period, to hopefully end up surveying like that exact location multiple times. So I'm going to say no there, and yes, I'd be happy to have my information shared. How did you hear about the Bumblebee Atlas? We'll say I heard about it through Master Naturalist program. Certify that you're 18 or over. There may be risks. And then read this trespassing policy to make sure that you're following laws and not trespassing. Then I need to acknowledge that I'm a registered volunteer and will not be getting paid in exchange. So yes. Then I go ahead and submit, and I will get an email confirmation saying that I adopted that site. The other thing I wanted to show you for the Pacific Northwest, so we can go over here. You can say find your atlas, explore Pacific Northwest. And we'll scroll down. And then I'm going to say, if you'd like to see what grid cells and time periods have already been adopted, you can visit this page. That's then going to take me to this long-term monitoring dashboard, which is a neat way to get a sense of what sites have already been adopted and during which time periods. And that doesn't mean you can't adopt that site. So if you really want to go to Idaho 1, just because 2 people have signed up doesn't mean you can't go. We would really appreciate as much data as we can. But we are trying to fill gaps. So if you're thinking, should I go to a site that's already been adopted or one that hasn't been, this way you can see who's adopted what in which time period and potentially go there. You can also look for gaps. So for instance, if you're thinking about Idaho 19, you could say, oh, well, I can fill these 2 in and I can coordinate with the person doing those other 2 time periods. I'm going to hop back to our presentation now that I've showed you how you adopt a site. So when selecting a site to survey, you'll also want to think about when to visit the site. The overall survey window for bumblebees is generally June through September, with timing of peak bumblebee activity varying by elevation, latitude, and other factors. In order to survey, you'll need to pay attention to factors impacting bumblebee activity, including temperature, wind speed, and precipitation. The temperature should be between about 60 and 90 degrees, with wind speed below 15 to 20 miles an hour and no precipitation. Bumblebees are pretty hardy animals and will fly even if it's fairly cool and overcast. So like I said, we'd ideally like to have each site surveyed 4 times throughout the summer to allow us to track changes over time and also increase our likelihood of detecting every species present at a site. The 4-hour time windows for survey time should be spaced out with multiple weeks between each survey. The 4 target time windows are June 1st to June 23rd, June 24th to July 14th, July 15th to August 4th, and then August 5th to August 25th. The goal in selecting a site is to select habitat with resources that will support bumblebees throughout the entire summer. To do this, you can look for a site that has a variety of habitat types. You'll want to pick a site that has some flowers, but also looks like it will have flowering resources throughout the summer. Some helpful questions to ask yourself on your first visit to the site are, do you see plants in bud that haven't yet flowered? So that's a good sign. Are there sites that are currently too wet but are likely to dry out? The whole site shouldn't be that way, but if some is, that's a good thing. Is there a permanent water source or maybe even melting snow that will help flowers bloom all summer? If so, that's a good sign. Are there many flowers already going to seed? That may be a bad sign, but also pay attention to the 3 questions listed above. Are you still seeing some queen bumblebees? If so, that's a good sign. It indicates that some species are at or near the beginning of their active season. And then finally, are there already males present? If so, that indicates some species are already coming to the end of their colony cycle, and that's probably a bad sign. You can learn more about participating in the Bumblebee Atlas by watching the other videos in this series. You can access those videos under the Resources tab on the Bumblebee Atlas website. You can also access additional resources by visiting the resources section. Okay, I'm going to hop back in here, and I just managed to escape out of all my windows. So bear with me for a second. We'll enter full screen. Okay. Also, my neighbor is weed whacking right now. They started right in the middle of that, so I apologize if you're having to listen to that. Hopefully it didn't come through. So one update since that segment was recorded, I just wanted to update the days of the week that the survey should happen throughout the season. So the dates are the same as last year, but obviously the days of the month are different than last year. So this graphic, this calendar just shows you what those weeks are. And this information is on the website as well. So you should be able to hop over and see that there. Oh, it looks like Rich, I think this one didn't get moved if you want to hop on.
Yeah. I just recognize, and it became true to me again as I was watching the video that Molly produced for us, that there are a number of different website resources, and some people tend to get confused. And we're sorry about that. That's just the nature of our program. We have one site, the bumblebeeatlas.org site that Molly was just walking you around, which is mostly an informational website where you'll find project resources, training materials, you'll learn about how to submit data, and a whole bunch of other things. That's sort of your information portal, is bumblebeeatlas.org. You do not need a login for that page. There's no password necessary. There is an option to log in. It's mostly for those of us that are editing the website, or that we have a few features that are only available for like project admins on the site. So there is a login option available, but you do not need to log in order to access anything on the website. Anything that you need to access, you should just be able to access free and clear without logging into the website. So don't worry about having a login for bumblebeeatlas.org. BumblebeeWatch.org is where you will submit all of your data once you've gone out and conducted a survey. So you will need a login for that site, and we'll talk a little bit more about submitting data later in the webinar, but that is the portal or place where you will submit all of your data, and you will need a login. That is the login username that you'll enter when you adopt a grid cell, as Molly just described in that video she shared with us. And then finally, Xerces.org is a great source for conservation information. If you need plant lists or want to explore other parts of the work that the Xerces Society does, including our podcasts and blogs and plant lists and habitat restoration guidance, you can find that at Xerces.org. But that is not directly related to the Bumblebee Atlas. That would more mean I want to go the next step and start acting more on conservation. And that's the purpose of the Xerces.org website. So those are the three websites that are affiliated with this program. BumblebeeAtlas.org is your information portal. You do not need a login. Bumblebee Watch is your data submission portal. You do need a login. And Xerces.org is your I want to take a next step website.
Great, thanks, Rich.
Yep.
I think I just had one more slide and then it was going over to you. No, I guess I think this is you.
Yeah, I think it was in addition to me there.
Sorry about that. Do you want to switch screen sharing so you can
Yeah, I'll take over
see your slides? Okay.
Yeah. Let's see. Okay. Molly, are you seeing my screen?
I think so. Will you click to the next slide? It looks the exact same as my screen, so.
Yeah. Did it switch?
It did not switch for me,
Okay.
but
it looks like it did to me. Like I can see what I'm sharing with other people, and it looks like it did. So I can see it changing. Yeah. Will someone just? Yeah. I think we're good. Will someone just say in the chat if they can see my screens changing?
Okay, looks like we're good.
We're good. Okay, great. All right. So good. I think we're good now. I did just want to, Molly just walked us through how to select and adopt a grid cell, which is fantastic. Now I'm going to walk you through sort of the next steps in the protocol. But before I do that, I do want to give just a quick overview of the Bumblebee Atlas website. Molly showed you where to find the adopt a grid cell and a few other things, but there is a lot of information on the website. We used to have like a Pacific Northwest Bumblebee Atlas specific site, but it's been integrated now into the larger website. So I just want to make sure that you're aware of where to find all of the different portal pieces that you'll need to participate in the project. So this is the homepage where you'll go when you go to bumblebeeatlas.org. You can explore the data from all of the years of the project using this dashboard here. If you want to find events, so if you're interested in like that ecology and conservation webinar, obviously that's already happened, but we have other webinars that are happening. You can click this events page and you'll see our online events here, like the Pacific Northwest Bumblebee Atlas that we're currently participating in is on here, and you can register for that here. You can also scroll down and there's a map of in-person events that we're having. You'll see that there's an in-person event that's being hosted on the Olympic Peninsula. So if you want to like see that, you can learn more information about that by clicking on that link. And then there's a registration URL down here at the bottom that you can click, and it will take you over to the registration page for that webinar. Also, if you go to your your get involved page here, this is where you'd find that adopt a grid cell page that Molly showed you. If you click find your atlas, it will take you to the Pacific Northwest page, or you can select the Pacific North page, and this is where you'll see Pacific Northwest specific information, including the dates that you can survey. There's also the link to that dashboard that Molly showed you about who's or how many people have adopted which grid cells and in which time windows. So that's where you'd find access to that. And then if you hover over this resources page, this is where you'll find the train online information. So you can find the videos that Molly shared with you and will share with you throughout the night, as well as where you'll find a copy to this webinar once we've adopted it. So if you select train online and then scroll down and click on Pacific Northwest, it opens up all of these different options that are specific to the Pacific Northwest, as well as some of our general guidance, which is further down on the page. Then if you go to print materials, you'll find the necessary information like your data sheets and other things. So at the very top of the page here, you'll see the data sheet and then an overflow data sheet. You all want to use this data sheet on the far right, the long-term monitoring data sheet where it says PNW only. This is the data sheet that we want you all to be using because it has the additional fields that are part of the long-term monitoring. And then if you scroll down a little bit further, you will see the Pacific Northwest specific information where you'll find the handbook, which has all the updated protocols on it. It has the Pacific Northwest Bumblebee Atlas specific data sheet. And then there's the Bumblebee ID guide for the Pacific Northwest, and then a placard for your car. If you want to have that while you're parked at a particular location, you can print that out and fill out the information and put it on your dashboard so folks know why the car is there. And then there's also some materials from the first phase of the Pacific Northwest Bumblebee Atlas down there as well. So that there are other resources available on here, like what equipment you'll need, and the protocols are on here. But for the most part, I think those are the resources that you will be using most. And I just wanted to make sure you knew how to find them on the website. So there you go. Those are the few websites that we have available to you and where to find stuff on the Bumblebee Atlas website. So the next step after you've selected and adopted a survey site is to review the survey protocol and to gather materials. And I just showed you largely how to do that. I'm just going to walk you through now on a more step-by-step basis. So you're going to use that resources tab on the Bumblebee Atlas website. And if you choose that print materials, this is where you're going to find the print materials that I just showed you. The Bumblebee Atlas handbook, the data sheet, and the ID guide are all on that page. The supplies that you're going to need to participate in this project are listed on the slide here. You're going to need some sort of insect net. We can provide you with some resources of the best places to find a net. There are some links on the website that give you options. And there have been a couple of new insect collecting equipment websites that have popped up in the last couple of months, and I'll try to remember to drop those websites in the chat here in a couple of minutes. There's relatively new resources. You'll need the data sheets that I just showed you on the website and some pencils, a clipboard. You'll need vials. You'll probably need somewhere around 20 vials would likely cover an entire survey. Some of you may need a lot more than that. Some of you may need fewer than that, depending on the habitat type that you're working in and the number of species and a whole lot of other things. But 20 is usually good to cover an entire 45-minute survey. And then you'll need a cooler with crushed ice that you will use to initiate cold anesthesis for your bees, to chill them out so that you can take clear photos of them. And then you'll need a camera. So you can use a dedicated camera or your smartphone. The cameras on them are good enough as long as you know how to use them. And then using, the plant and bumblebee field guides to help you identify the bumblebees and the plants that you find. And then some way to find specific locations. You can use your phone for this, or you can use a GPS unit if you have one. And then you can use a timer. And your smartphone can also obviously work as a timer. So a smartphone is a great tool here. It covers a lot of bases that you can need. There are also apps available for for plant ID that you can also have on your phone. So lots of this can just be covered by bringing your phone with you on your field in the field. For vials, we recommend, I find that the 30 milliliter centrifuge tube is really the best size. And you can see the pictures on this page will show you roughly what those look like. I like the 50 or the 30 milliliter ones. The 50 milliliter ones are a little bit easier to find. But if you search on a search engine or on one of the big box store sites, you can just Google the 30 or 50 milliliter centrifuge tube and you'll get options. You can also use things that you might find around the house. Prescription tubes, do make sure to clean those out thoroughly before you use them. You can use baby jars. You can use all kinds of things can be used. So don't feel like you need to go buy something, but there are relatively affordable options out there. It's really important that we sanitize our equipment, mostly the vials in between every survey. We just don't want to be, as you know, if you've been part of this project or if you watch the ecology and conservation webinar that we delivered, disease is one of the major issues in bumblebee decline, and we really want to make sure that we're not the vectors moving diseases around the landscape. So we want to do our best to protect the animals we're trying to learn about, and cleaning our equipment is one of the best ways that we can do that. So in between each survey, make sure to wash your jars. You can use warm soapy water or a mild bleach solution, a mild alcohol solution. Any of those options can work. It's also a great idea to sanitize your net. And again, you can use warm soapy water, just rub it around in soapy water, rinse it and let it dry. Or you can spray it with a mild bleach solution, and that works just fine as well. So please sanitize your equipment. And then, yeah, having a camera are the options you're going to need. The most important thing, really, any camera can work. The most important thing is that you practice and learn how to use it before you head out in the field. If you've participated in this project for a long time, you will know that taking pictures is really the most important part of the job here. If you go through all of the effort of selecting a survey site, driving to the survey site, conducting a survey, and then take photos of the bees that make it difficult to identify them, a lot of that effort hasn't been as productive as it could have been. So we really want to make sure that we're taking high quality photos and allowing those identifications to happen. We highly recommend the Olympus TG model cameras. They're compact, they're waterproof, they're shockproof, and they take fantastic photos at very close range. Other good options other more affordable options are to buy these clip-on lenses that you can attach to your iPhone or your Android phone. As you can see in the lower right-hand corner there, it's a little clip-on, and it turns your cell phone into a macro camera, and you can take really detailed photos that way as well that are high quality. But again, please, the most important thing is that you just practice with whatever equipment that you have and learn how to use it before you're actually in the field, and only have a few minutes while a bee is warming up to learn how to do it. So take your time to learn how to use your cameras. The cooler that you need is like a lunch-sized cooler filled with crushed ice is the most important thing, or about the right size. It's really important that you not use the blue ice options, particularly the newer ones by like a company like Yeti. They are formulated to chill down to like 30 or 50 degrees below Fahrenheit, which is just too cold for bumblebees. So if you have a chest freezer with one of those in it, the temperature of that is going to be so cold that it could actually kill a bumblebee. So please do not use those, at least for direct contact for the bees. You could put them at the bottom of your cooler and then cover them in crushed ice, and those would help keep your ice cold throughout the day. But please don't use just those blue ice options, as we've had mortality in the past from those. So please use crushed ice. Cubes are the best way to go. Like from a gas station, just get a bag of ice. Those work really great. Yeah. And then you can find recommendations for equipment on the website. If you go to resources and just hover down to equipment, you'll see a lot of the information that I just shared with you if you need a refresher. The data sheets, again, we showed you where to find those on the website. This is what they look like. You're going to need to bring one of these for every survey that you're doing. So if you're doing a point survey, you would bring one of these. If you're doing a roadside survey, you would need to bring 5 of these along with you. So make sure that you know what kind of survey you plan to do and make sure you have enough data sheets. Bring a couple extra just in case, especially with ice and water around, you never know what's going to happen. Just as a little pro tip here too, I will also say one of the things that I make sure I do at the end of every survey, once I've filled out my data sheet, is I use my camera and I take a photo of both sides of my data sheet. That way, if I lose my data sheet or it gets destroyed by water or my dog eats it or whatever, I've got a backup of it on my phone and I haven't lost that important data that we've collected. So that's just a pro tip for handling data sheets after you conduct a survey. So when you get to your survey site, you know, what you'll do is you'll fill out the top of this data sheet with the information that's important to your site. So, you know, the site name, and you can call it whatever you want, but you will need a site name when you enter the information on Bumblebee Watch. So do give it a name. The site ID, you can write down here the date, the latitude and the longitude, and please use decimal degrees, not in minutes and seconds. The website will not take minutes and seconds, and it can be difficult to convert. So please try to enter the data here in decimal degrees. The number of observers, observer name, and then you're going to fill out information about what kind of survey it was, how large it was. A standard point survey for a Bumblebee Atlas survey is 1 hectare. So most of you will be entering 1 in the survey area here. And then what method you used to capture bees. Some of you are going to capture all of the bees that you find. Some of you are going to only look for different bees. So you're good enough at Bumblebee surveying and you recognize when you've found a species, or if you know, the idea for this program is to really try to collect diversity of bees. And so we want to get every species that's present at a point. And if you are confident enough in your ID, as long as you're getting enough of lookalike species that will capture like a Vosnesenskii and Caliginosis complex, then you could focus on collecting different bees so that we're getting every species present at the site. Or you may do some combination of that, where you start by collecting all of them, but you're just getting gobs and gobs and gobs of what look like Bombus vosnesenskii. And so then you're going to start focusing on diversity. And that might be an area where you circle combination here. The start time, the end time, the number of survey minutes. Again, a standard Bumblebee Atlas survey is 45 minutes long. So most people should be entering 45 minutes in this site. If you're doing a points or a roadside survey, you would be entering 15 minutes at this point. And it's important to recognize that this is in person minutes. So if you had 3 people, if you had 3 people that were conducting a survey, each individual person would survey for 15 minutes, and then the total would be 45 minutes. So your start time and end time might be only 20 minutes, but the total survey time would be the number of surveyors times the time that each surveyor surveyed. So you'd still enter 45 minutes in this survey minutes block. And then you're going to get some basic weather information. You can either do that, you know, in real time on your phone using a weather app if you have cell service, or you can do it later when you get home and just look up the data at the time when you were conducting your survey. And then there's some information about the habitat, and you're just going to fill that out. We're going to go over that in more detail later here. So I'm going to skip over this and move forward. And then we're going to ask you about the plant species that were in bloom at the site. And what you're going to do is you're going to select up to 15 of the most common blooming plants that you find at the site. And then you're also going to estimate what proportion of the site or of the blooming plants that those individual species made up on your of your plot. So I'll go over that in a little bit more detail. I have a few more photos. I probably didn't say that very well. But basically what you're going to do is you're going to write down is up to 15 species. You don't need to collect more than that. 15 would be the maximum. You're going to write down their names on this sheet, and then those are going to make up 100% of the flowering resources. And then you're going to estimate what proportion did each of those individual species make up of the whole of the flowering resources at the site. And you're going to enter that information, which is why you see the 60, 25, 15 here. I have another example I'm going to share with you later that'll help make this a little bit more clear. And then lastly, after you've captured all your bumblebees, each jar that you capture will become a line on your data sheet. So you're going to enter, you don't have to enter, like if you think it's the sex of the bee, you'd enter in this column. If you know the species or you think you know the species, you would enter it in this column, and then or in the second column there, the bumblebee species, and then you're going to enter its host plant in the third column, and then the photo numbers, and you would pull this from your phone or from your camera of which photo numbers are associated with each observation here. And then you'll notice also that there's a FD 110 here, and I'm going to go over that in later detail as well. But what we want is when you detect one of our two species of conservation concern, Bombus occidentalis or Bombus morrisoni, the western bumblebee or Morrison's bumblebee, we want you to note how long or what time of day, so you have the start and end time on your data sheet. We want to know how far into your survey did you find that bee. So really try to, and some of you may not be able to do this. If you can't, it's okay. But if you know those two species well enough and how to identify them, especially with the western bumblebee, it's incredibly helpful for us to know how long into your survey that you found that species. So you're going to write your time of first detection on your data sheet for that bee, and then enter that later into the database so that we can use it in our analysis. And then at the very bottom of the data sheet, you're going to enter the volunteer data about how much time and effort you put into doing this, and thank you. Generally speaking, a point survey is a 1 hectare plot. So it's about 2.5 acres. The size of a soccer field is generally about a good estimate for how much area you'd be surveying. It's 45 person minutes as a reminder. You're going to include that habitat data like we just talked about. And then each bumblebee you're going to catch, chill, photograph, and then release as you record them onto the data sheet. So again, just as a reminder, and this is a little bit different from the first phase of the Bumblebee Atlas project, is that we're really focusing on diversity. We want to try to capture as many different species as we possibly can. So keep your eye out for bees that look different, and you're welcome to move around and capture those bees instead of just capturing every individual that you find at the site. If there is a look-alike, like I mentioned with the yellow-faced bumblebees, do try to catch at least 10 of them so that we can capture any species that may look similar in the field, but are actually different when we get to the ID portion of this. I talked about the time of first detection. This is for western bumblebee and Morrison's bumblebee. If you're able to identify those species in the field, or once you get it in the jar, please note the time of first detection. Take a voice memo on your phone, write it down on a notebook so that you can enter that data later when you process the bee. And then another difference from the first phase is we want to know more information about honeybees on the landscape and whether they're impacting bumblebee communities. So what we want you to do is after you conduct your survey, set your timer for 10 minutes, and we would just want you to walk around the site kind of casually and just count the number of honeybees that you see in that 10-minute period, and then enter that number on the data sheet. And then when you go to fill out your data later, you're going to enter that information on there. So please make sure to do that honeybee count after your Bumblebee Atlas survey. It's just 10 minutes. If there's two of you, you can each set your timer for 5 minutes, go count how many honeybees you see, and then just combine your numbers at the end and do it that way. And then, as I mentioned, you're going to document the plant community, list up to the top 15 plants and the percentage of how much of the site that they're making up of. And this is just kind of an example of what that looks like. So if you think about the square here on the left as your hectare, and these are the flowering patches that are all around. You can see I have, 9 different species of flowers on here, and they're sort of separated or disparate across the region. So what the first thing I want to do is count the number of blooming species that I have, and I want to write down the name of each of those species. And then also one of the things that you're going to do is try to estimate how much of the survey area was covered by those plants. And we're not really asking like a percent cover question here, like how much of the site is covered with flowers. It's more in how much of the site could you actively search for bumblebees on flowers. So in how much of the site were you surveying? Basically is the question. And how much of it were you walking between flower patches? So you can see if I move all of those flowers sort of to the left-hand side of the plot, as I was able to do, because this is a cartoon and I'm able to move those around, you can see that roughly about 50% of that site was covered in flowers, where I could reach a bumblebee on a flower in a net, is basically what the question is. And so when you get that question on the data sheet, how much of the survey area was covered in flowering resources, that's where you answer this question of 50%. So in this case, it would be 50%. You might be in a site that has less, you might be in a site that has more. And then what I want to do is I want to say, okay, that 50% of the plot, but that 50% of the plot is still 100% of the flowering resources. And then what I'm going to do is I'm going to look at the distribution of those, in this case, 9 different species of flowers, and estimate what percentage of the flowering resources did that individual species make up. So you can see this sort of peach colored flower that I called flower 1. I estimated that made up 5%, and the same with the purple aster type flower. The blue aster type flower is a little bit more, so I gave that 8%. And then the pink flower is about the same as the blue, and then the purple is a little bit more. And then I add all of these up, and once you're done, these should all add or sum to 100%. And you're just gonna do your best to estimate this. It's okay if it's not perfect. You don't need to throw quadrats around or guess. We're just trying to get some idea of what the most common flowering plant species out there. So please do your best to estimate this. Again, it does not have to be perfect. We just want you to do your best. And the ability for us to be able to quantify the flowering resources just gives us a lot more power to look at what bumblebees are truly selecting versus what's available to them. And this allows us to build better plant lists and understand more what the bumblebees are using when they're out on the landscape. So this data is really important for us, but don't stress about it. Just do the best that you can with the time that you've been granted. And then you're going to enter that information on the data sheet. So again, I had all of those 9 separate plants. I enter them on the data sheet. And then I include the percentages on the data sheet of how much they make up. So I took plant 1, 5%, plant 2, 5%. You can see that as I entered it here on the data sheet. So that's what you're doing in this section. This is new for the second phase of the Bumblebee Atlas. We also did it last year. So if you did it last year, you're old hat, but just want to make sure to clarify this is a new program if you're new to the long-term monitoring protocol. So that's more or less the data you're going to end up on your data sheet. I now just want to spend a few minutes talking about photography. So again, the most important part of your job as our volunteers is to be a photographer. Of course, it's wonderful to go out and catch bumblebees and chill them and do all of that wonderful stuff. But again, if we do all that and take pictures that aren't good enough for identification purposes, then a lot of that effort has been wasted. And we don't want to waste anybody's effort. So please take the time to practice. If you need help learning how to use your camera, both Molly and I are available to do a Zoom call with you. Or if you're here in Portland, feel free to come into the office and we can help you with the technical aspects of your camera. I know that it's not easy for some of you, but we can do it, and it just takes a little bit of training and practice. And that's another thing that you can do is reach out, like on the Facebook group, if you're struggling to learn how to use your camera. There's a whole host of highly qualified volunteers out there that have been doing this for a long time and are really good at it. And I know a lot of them are willing to lend their expertise to you about the best ways to use the resources that you have. So if you need help, please reach out to us. Taking pictures is really one of the most important things that you can do. And generally speaking, I'm just going to go through the photos that we hope that you take as you document each bumblebee that you've captured, put in a jar, and then you're processing. So we need a photo basically to show all the parts of a bumblebee. You can see over here on the lower right, basically the anatomy of a bumblebee. You've got the head, the thorax, and the abdomen. And the abdomen is made up of 6 tergal segments. And we want photos that basically show all of those parts of the bee. That's basically all there is to it. And the most important thing is when you take a photo, what I want you to do is to look at it after you've taken it. And if you can see individual hairs on the bee after you've taken the photo, it means it's sufficient quality for us to see the features that we need to see. So look for that ability to see individual hairs in each photo that you take. If you can't see individual photos, it means that we need to work on taking higher quality photos. So just use that as a guide. Can you see individual hairs? As you can up here in the upper right-hand corner, hopefully you should be able to see the individual hairs on that bee. And you can see, I can also see the very front of this bee's face. I can see the hairs on the front of the bee's face, and I can also see the colors of the hair on the top of the head. So those are the pieces that we need to see on the face. It's also really important to see the cheek, and we don't show that in this photo here. I have a photo of that later that I'll show you that we also need to see as part of the head. And then for the thorax, we want a photo that shows the part of the thorax in front of the wings, the part of the thorax between the wings, and the part of the thorax behind the wings, and the part of the thorax below the wings. All of those parts are really important to for us to be able to see. I'm going to try to do a little annotation here and see if this works. So this portion here in front of the wings is what we want to see. That's one part. This is the portion between the wings. And here's the portion behind the wings. And then here's the portion below the wings. So we want to be able to see all of those different parts. So it's ideal if you can take a photo from directly overhead, looking down, and then one photo from the side, like this one, allows us to see all of the features that we need to see. So those are the photos for the thorax. I'm going to try to clear all of these now. Okay. Oh. Okay. And then for the abdomen, we want a photo that shows every single segment on the abdomen. So like for here, you can see down here is where we show you the breakup of the different segments on a bumblebee's abdomen. And you can see here on this photo, there's the end of T1 is right there. There's the end of T2, T3, T4, T5, and then T6 is down there at the point. So this is an excellent photo of a bumblebee abdomen. Most importantly, the wings are spread wide so that I can see the middle of all of those segments. If the wings of this bumblebee are folded over the back of the abdomen there, I can't see the features that I need to see in order to identify it. So it's really important you can use forceps or your fingers or a pencil to pry those wings apart and hold them apart while you take the picture. It's okay if the forceps and the pencil are in there. The most important thing is that we're able to see this region of the abdomen because there's some key features for a few species that we need to see those segments for. So please make an effort to take a clear photo of that section. And then, just again, please make sure that the bumblebee is in focus. On the photo on the left here, you can see that it's just, you can't see enough detail from a photo like that to identify the bee. The photo on the right is great. You can see those individual hairs on the bee, and that's what we are striving for. Likewise, try to play with the white balance and make sure that they're light enough. If you take pictures in in the shade or in direct sunlight, it can really impact this. So getting experience with your camera in different light conditions is really important. And again, practicing at home before you need to, or practicing with an object that's not a bumblebee while you have your setup is a really important thing to do as well. And then here's that photo of the cheek that we need to get. So when you're taking a photo of the head, if the bumblebee has its face down like this, it's really, like you see on the left, it's really hard for us to see the features that we need to see. But over here on the right, we've put the bumblebee on its back, and you can take a photo from an angle, and then we can see the cheek of the bee. And what we're trying to see is this ratio here of how wide is the cheek compared to how long it is. And that's a really important character for us to distinguish between some of the close look-alike species. So the first photo that I always take of a bumblebee is I put it out of the jar on its back, and I take this photo head on of the bee. That shows me the hairs on the face. It shows me the hairs on the top of the head. And then I also get this view of the cheek. So that is the first photo that I take while the bee is completely, anesthetized and on its back. It's a great time to take that photo. So that's my clear recommendation for taking that photo. And then also make sure, it's really important that we try to keep the bees dry. It's good for them to stay dry because they don't swim. But it's also, it's really important for identification to be able to not have matted bees where you can't see individual features. So make sure that your jars that you're using are sealed. Make sure they're closed tightly. And then also make sure that you're not putting the lid of the jar below the water line. So feel free to empty the water out of your cooler and just use the ice cubes. Because if they get submerged underwater, you get bees that look like this. That's not good for the bee and also not good for identification purposes. And again, here's just that picture of folks prying those wings apart so that we can see that feature in the middle of the abdomen. And a good example of why we need this is just that photo on the right here. This is Bombus griseoculus, and you can see this crescent of brown that it has in the middle of T2 is a feature that we might miss should that bumblebee have wings folded over its back. So really important to get those wings out of the way and take those nice clear photos. It's fine if there's other things, like these are not National Geographic photos that we're looking for here. We're just looking for photos that show characteristics. So if there's a pencil in the photo, or there's a flower part, or your fingers in there, or you can see your kid's face in the back, like it's all fine. It's good. Don't worry about it. Other things can be in there. It really is, just try to get the photos that we need. Don't worry too much about making them museum quality. And then the final photo tip is please don't take photos of bumblebees through jars to submit for identification purposes. If you already have a bumblebee in a jar, please put it in a cooler. And then once you have it in the cooler, take the bee out of the jar to take those photos. You can't see enough through jars. The light gets abstracted, the view gets weird, and they get scratched up. And it just is not a great way to be able to identify bees. So please take your bees out of the jar before taking photos. And then when you go to enter your data, so now we've sort of processed all our data. You've taken those super high quality photos. We want to make sure that you're entering your data in the right place. In the past, when you went to go submit data to Bumblebee Watch, you used the Bumblebee Watch project code, or the Bumblebee Atlas project code, which you'd see here on the right. For the Pacific Northwest, we would like you to not use the Bumblebee Atlas code because that doesn't have the honeybee field or the flower percentage fields that are available that you're collecting when you're out in the field. So you're going to use this lower part of Bumblebee Watch where it says or select from your regional projects. You're going to click on that area. And then once you've clicked on that area, it's going to bring up this menu, and you're going to select PNW Bumblebee Atlas. And that's going to bring up the form where you're going to fill out all of your data. And you're just going to match up your data sheet with the data fields. And we made an effort this off season to make sure that the data sheet is 100% aligned with the data fields on the website. So any confusion that you may have had in the past should be cleared up. So anyway, that's just a way to say, please use the PNW Bumblebee Atlas project code when you're submitting your data. Likewise, there isn't a place on the website which explicitly shows you how to or where you're going to enter this data of the time of first detection for the bee. You need to enter that into the notes portion of the Bumblebee when you submit that Bumblebee sighting. So when you go to upload your Bumblebee, you're going to come to this field, and if you're entering a Western Bumblebee or a Morrison's Bumblebee and you happen to know that and you collected the time of first detection, and the observation notes where you submit your data, this is where you're going to enter that time of detection, and you're going to put the time of detection right there in that field. So it goes into the observation notes of the individual species is where you're going to enter those data. Just so you know, we now have connected, it's a new upgrade over the winter time where we've now connected Bumblebee Watch to an external database to standardize all of the floral data that you'll be entering into the website. This should greatly expedite your data entry process, and it also standardizes things on the back end and makes it easier for us. So you'll notice here that under Floral Host, you'll see that there's a very, it's a very sort of official looking thing. And that's because when I entered this data, I started typing Potentilla gracilis, and then it popped up options. I select Potentilla, and it automatically entered this information, and this gets carried on and entered into the database. So you're going to start typing just as you have in the past, and it kind of works as a search tool, and then it'll pop up options. The more you type, the closer the options that are pop up will match your selection, and then you'll select that option. So you can do that with scientific names, like you know that was Potentilla, you can start typing that. But if you just knew that this was cinquefoil, you can also just start typing cinquefoil, and it will bring up all of the related options as well. So it works for common names and for scientific names. Please do your best to enter only the level of information that you know. So if you know that this is a cinquefoil, but you don't necessarily know that it was Potentilla gracilis, you should be able to enter that information in there as well. There should just be a genus option that you know it was a cinquefoil, but not that you don't know that it was slender cinquefoil. So please make sure to only enter the level of information that you are certain of when you enter those data. And then I just wanted to let you know that we, over the last winter as well, have been collaborating with an organization, or really a person, called Bee Machine. This is Brian Spiesman. He's at Kansas State University. He developed an AI bee identification tool using the Bumblebee Watch dataset and other resources that are out there. We are implementing this AI tool into the workflow for you. So you'll be able to use this to identify the bumblebees as you submit them. We're still going to look at every single bumblebee observation that comes in. Molly and I will be the two people that will do that. But this tool will be available for you as sort of an educational tool. As you're submitting data, you can see what the AI tool thinks that it is. And we'll share some more information with you once that is officially live. We're hoping that it's going to be live on the website by June 1st. Lastly, I just wanted to share with you, I did mention this a bit earlier, but we are having an in-person field training on the Olympic Peninsula near Hoodsport, Washington. It's going to be Saturday, June 7th from 12:30 to 3 p.m. I'll be there on site. The main purpose is to give you hands-on experience collecting bees and taking pictures. So if you need help or want help with any of your equipment, and you live close enough where you can come join me, please do stop by. I'd love to see you and meet you in person and interact with you, or see you again. It would be really fun. If you want to register for that, you can do that on the events website that I showed you earlier. This QR code will take you there, or you can type in the URL that's up there in the upper right-hand portion of the white part of the screen, and that will take you there as well. And then one last update here is when Molly showed you that video of adopting a grid cell, there was a question of, can we provide your email information to other volunteers so that you can coordinate surveying the same within one of those hexagonal grid cells? What we're going to do is we're going to use that information to set up a Google group so that you can directly email other volunteers and coordinate with them. So if you say yes to that question, I'm going to add you to this email Google group where you'll then be able to participate and reach out to other volunteers and look for help within your grid cell or coordinate with other volunteers within your grid cell so that you can do this in real time and you're not dependent on Molly and I trying to help you, because we're so busy during the field season that it's really hard for us to respond to those emails when they come in. So we're trying to create a space where you can all help each other and coordinate with each other. I don't expect that there's going to be gobs and gobs of emails coming through this. It'll just be a few emails. And when you sign up, we'll share some information about how you can control how often this information would come into your inbox. You can shut it off so that it only shows up when you go look to check on it, or you can get every email, or you can get daily digest emails. So this shouldn't overwhelm you with information. And I think it'll be an effective way for you to coordinate with other volunteers within your hexagonal grid cell. So stay tuned for more information on that. We should have it before the start of the field season. And with that, I think I'm going to turn it back over to Molly.
Yeah, thanks, Rich. Do you want to just play this video or do you want me to take over screen sharing?
Oh, sure. I can play this, although were you going to play the whole thing or were you going to stop at some point?
I was thinking we'd play the whole thing, but if you think part of it's more relevant than all of it, that's totally fine.
No, I just wanted to make sure. I can play it since I'm already sharing, and then we'll switch after that.
Sounds good. Thanks.
Yep. Hi, my name is Rich Hatfield. I'm a senior conservation biologist for the Xerces Society, and I'm here to teach you how to do a survey for the Pacific Northwest Bumblebee Atlas or the Nebraska Bumblebee Atlas. They both have pretty much the same survey protocols. So I'm here today. I'm in a beautiful natural area. It's a nice, warm, sunny day. I've got my net and I have some vials in my pocket, so I'm pretty much ready to do a survey. The first thing you want to do is make sure that the site is big enough. It needs to be about a hectare. And a hectare is about the size of a football field, lengthwise, square. So sort of a football long and the same distance wide. It's 100 square meters. So a football field, including the end zones. Another way to think about it is the infield of a track. If you ever ran track, the entire infield on the inside of a running track is about a hectare, or a soccer field is also about a hectare. So you want to make sure it's big enough. And then usually what I do once I decide that it's a big enough site is I walk around and I make sure that there's enough flowering resources. You want to make sure that there is something that bees could be visiting. Otherwise, it's probably not worth really conducting a survey, or maybe come back at a later date when there are flowers. One of the things you want to do when you're out pre-walking your site is make sure you're keeping an eye out for inconspicuous flowers. Sometimes flowers will grow down amongst the grass. They can be important for bumblebees and often are. This plant is called self-heal and often grows underneath the understory or overstory of grass, and it's a great bumblebee plant. Another important thing when we're out looking for bumblebees is we want to make sure we're looking at all blooming plants. If I was standing here looking in this patch, I might be drawn to the violets, the dandelions, or maybe to the bluebells over here. But in reality, one of the most attractive plants in this patch has green, inconspicuous flowers, but bumblebees love it. This is a plant called waterleaf,
And the flowers aren't quite open yet because it's still very early in the season here. But these little green guys here are going to bloom into little, beautiful little flowers that hang down sort of under the understory. And they often can't be seen. But bumblebees like Bombus flavifrons and other long-tongued species really love these flowers and will visit them. So while you may not be drawn to them with your own eyes, it's really important to pay attention to what's blooming and making sure that we're spending time looking at all the flowering resources on the landscape, looking for different species of bumblebees. Other things you may want to look for as you're doing a pre-walk around your site are things that may be a potential hazard. You may want to avoid plants like poison oak or blackberry, although it is notable that both of these plants do flower and can be attractive to bumblebees. You'll also want to look out for holes or other things to avoid, like electric lines, fences, barbed wire. Just make sure you're safe while you're doing your survey. You also want to keep an eye out for property lines to make sure you're not crossing from public land onto private land. And also keep an eye out for areas that you may want to spend a couple more minutes, areas with high quality forage that you know are especially attractive to bumblebees. Or keep an eye out for blooming trees, meaning don't forget to look up. There are sometimes flowers that are above your head that can also be attractive to bumblebees. Once you've finished your pre-walk, you're ready to start your survey. Materials that you want to make sure that you have with you before you begin your survey are an insect net, jars to place your bumblebees, a cooler with crushed ice. Make sure you have a timer. And then for processing your bees, you'll need your data sheets as well as a camera. We recommend having a dedicated camera, ideally with a macro function, but in a pinch, a smartphone can work as long as you learn how to control your white balance and take high quality photos with it. I typically set up a station in the middle of my survey area, which has the cooler and the area where I'll process the bees later. And then I grab a handful of vials and head out to go conduct my survey. And when you're conducting your survey, you want to make sure it's 45 minutes long, and that's 45 person minutes. So if you're doing it with a friend, you would do it for 22 and a half minutes. And if you did it with two other friends, it would be for 15 minutes. You just want to have 45 total minutes of search time. The important thing also is that when you're doing a survey, you want to pause the timer while you catch a bee. So it's 45 minutes of searching, not 45 total minutes. So you want to pause the timer when you catch a bee and while you're putting that bee into a vial and while you're putting that vial into the cooler. That doesn't count for your search time. The other thing that's important while you're doing your survey is you want to make sure that you're associating every bee observation that you have with a flower. So one of the important things about the Atlas project is we want to learn what resources that bees are using. So while you're out collecting bees off of flowers, you want to make sure to record that information so that we can gather it later and use that information when we analyze the data from the project. So with each bee that you capture, you want to also record the flower on which you captured it. Catching a bumblebee in a net is not an easy task. It takes a lot of practice. So one of the things that we recommend is that you spend some time practicing in your backyard before you go out and actually conduct your survey. That way you'll get some competency and some confidence so that when you see a bumblebee during your survey, you'll actually be able to net it. There are really two different ways to go after a bumblebee on a flower. One way is to just swing your net and try to catch it in there like this. The important thing is after you've swung, you want to make sure to turn your net over to close the bottom of it so that the bee doesn't come flying right out into your face. There's a number of ways to do that. I like to do a little flip, but the easiest way to do it is to swing and then just turn the net over 1-2 times. And then you'll have the bee in this section of the net that's laying over the top. The other way to go after a bee instead of sweeping is to just go over the top and drop the net on the bee like that. That way the bee will be trapped in the net The next step then would be to grab the bottom of the net and pull it up to the top, and the bee will fly right to the top of the net because it's attracted to the sun. Let's see if we can find some bees to practice on. So follow for her a bit. She looks like she's looking for a nest. They can be really hard to track. We can just throw the net over the top of them. That sometimes while they're flying, and I got her. I can see her moving in the net there. So what I do is I pull the net then up to the top and make sure she's under there. See her right in there, and then I can put her in the vial. Okay, so now that I have a bee in the net, what I need to do is get the bee from a net into the vial. And the way I'm going to do that, she's somewhere in this folded over area. You can see her crawling around right here. And so what I want to do is make sure she gets all the way down to the bottom of the net. So what I can do is I can just take the net and just go back and forth a couple of times and then fold it over. And then I like to grab it when I know she's above my hand here. So I know that she's between my hand and the bottom of the net. And then I can take my jar, take the top off, but I want to keep that cap close by. And so then what I do is I stick the jar into the net, and it goes through my hand here, okay, so that there's no way for the bee to get out. And then what I can do is just sort of move the net around until I'm sure that I have the bee in the net. And unfortunately, my net is very dirty, so it's hard for me to see. But I can hear her. I know she's in there. And usually you'll hear that sound, and you know she's in your jar. I know that she's in the jar. And now I can just use the net basically as the top of my jar. So I'm just going to reach my hand in, and again, I'm going to grab that plastic vial, and I'm just pulling the top of the net over the top of the jar, so the net is still serving as the top until I get to the edge, and then I'm just going to slide it directly into and put the cap directly on, and then I can close the top, and I can see the bee there in the jar. And that is how you transfer, or at least one method for transferring a bee from a net into a jar. Okay, so now I want to show you another method for getting a bee from a net into a vial. You can see the bees crawling around in the vial right here. And I'm sorry, in the net right there. And I want to get her into the vial. So one thing is a good thing to do. Some people just aren't comfortable holding the net while they do this, so they like to put it on the ground. But you can see if I hold the net straight up, the bee will fly all the way to the top of the net, okay? And then I can move forward. So some people just like to set it on the ground and then use the ground sort of as the cap, and then stick the vial into the net. And then again, same principle. You can pin the bee against the net as the top, and then you can stick your other hand in there and just put the cap on, and then you'll have the bee in the jar. Simple as that. And then what we want to do is we want to take the bee once it's in a vial, and we're going to put it into the ice pack. And it's ideal to stick it as far into the ice as we can, so that the ice is completely surrounding the bee, and then it'll chill very quickly so that we can take the photographs that we need to take. One of the things that I would do in this situation is I would take the flower that I captured this bee on, and I would put it into the jar so that later when I was processing my bees, I could know which flower that I caught this bee on. Keeping that keeping track of that is very important for the data we're collecting for the Atlas project. Or in this case, because this bee wasn't visiting a flower, it was just flying around, it's not going to have any floral association. So if I don't put a flower in there, then I'll know that when I process my data later. But if I'd captured this bee on, say, a dandelion, I could come over to a dandelion flower and just grab the flower off and pretty easily just tuck the flower into the jar with the bee. And then later when I process this bee, I'd have the flower and the bee in the same jar so that I can keep track of it. Other people like to take notes and have a notebook, and they will number their vial and say this would be bee number 1. They could go bee number 1 and they would write down the flower or take a picture of the bee next to the flower. There's lots of different ways to keep track of it. We don't care which way you do it. We just care that you do it. So when I finish my survey, I like to get a station set up to take my photographs. I want to make sure that I have everything I have and everything I need before I get started, because when I take the bees out of the cooler, they're going to start warming up, and I want to make sure that I have everything. So I want to make sure that I have my data sheet and a pencil that has lead in it. I want to make sure I can take my notes. And I want to make sure that I have my camera. The other thing I like to do is make sure that my camera is set so the pictures I'm going to take are appropriate for the lighting conditions. So I usually will take a cap of a vial or something like that and make sure that I have my camera in the right settings before I get started, so I don't have to wait while the bee is warming up. So I get it in my macro setting and make sure that the pictures that I take look good. So here I am, I'm going to take a picture. And that looks pretty good. See here. It looks pretty good. I could probably add a little bit of light. It's a little bit dark, so I may change the white balance a little bit, but for the most part, it looks pretty good. So once I'm ready and I've got the information that I need, I can start processing my bees. But first I want to make sure I've filled out the top of my data sheet. So I have my site name and my grid cell ID in here along with the date and the survey map.
Okay, I'm gonna stop sharing and we're gonna stop playing the video because we've largely gone over photos, and the data sheet that we show in this is different than the data sheet that we have available now, and I don't want to confuse you, so we're gonna move on and deal with the rest of this because I think we've covered a lot of this already in the webinar. So I'm going to turn it over to Molly and let her take us through the rest of the night.
Sounds good. Thank you, Rich. So the last step, once you've gotten in the field, you've collected all your data, is entering the data online. And that's really one of the most important steps, apart from taking really good photos, is getting everything correctly entered online so that we can identify it and your data can go into the big database and be used for science. So I'm going to show you, this is a recorded tutorial that we show you on the website, how to go on and enter your data. So we just have that, and then we had a little more content than anticipated, but we're still on track to be done by 7:30. And we'll have a little time for questions. But if you have questions, feel free to put them in the chat, and Rich and I will respond to those. This is the 5th and final segment in a 5-part training series. This segment provides an overview of entering your survey data online. Visit the Resources tab on the Bumblebee Atlas website to view the introduction segment, as well as segments on selecting a survey site, reviewing the survey protocol, and gathering materials, as well as conducting a survey. The final step in conducting a survey for the Bumblebee Atlas is to enter your data online. This allows us to confirm the identification of the bee. Your survey and associated bumblebee will also be entered in a comprehensive and publicly accessible database where scientists can access the data for use in research and conservation projects. To enter your data, you'll need your completed data sheet, photos of the bumblebees associated with that survey, and your Bumblebee Watch account login information. I'm going to walk you through entering some example data. Here's my example data sheet. I also have a folder on my computer with photos of the bumblebees from that survey. The file name of each photo lines up with the photo number on my data sheet. Now I'm going to go to bumblebeewatch.org and log into my account. And I'm going to switch over to a live view of the website so that I can walk you through this. All right, so here we are at bumblebeewatch.org. You hopefully already have an account from adopting a site. If somehow you don't, you can create an account up here. I'm logged in, so I could go look at my profile. I'm going to go back to the home page. All right, so what I want to do is add a sighting. We'll go to add sighting. Because this is part of a regional Bumblebee Atlas project, we want to select our regional project down here. And that's because our data entry sheet is going to be a little bit different than some of the other regions since we have moved on to phase 2 of long-term monitoring. So we're going to go down to Pacific Northwest Bumblebee Atlas. This is where you'll enter survey data. If you took a photo of a bee not related to a survey, you could select incidental here and just upload your photo to be identified. So that brings us to this data entry form. You can choose from your locations. So if you've entered data previously, your sites will show up in this dropdown. You can see I have a variety of sites. This is my first time recording data for this site. So I'm going to go ahead and enter a new location. But next time I survey at the same site, I will have it show up in that dropdown. So we're going to go to workshop demo. We'll type in our latitude. And we'll do our longitude. Make sure to get that negative. Otherwise, you'll end up in Mongolia or somewhere like that. Select your accuracy. So we're going to say, you can look up how accurate a GPS or phone unit is. I'm going to say like 25 to 50 meters. Probably a phone is smaller. It's like 0 to 20. Date of sighting. Say 6/1/2025. Oh, must be in the past or today. Let's see, I'm not recording this at that day, so we'll do March 13th. Survey type. We're doing a point survey. Like I talked about, the Sentinel sites, we're focusing on point surveys. If you've done roadside surveys in the past, you could select roadside here. But the main focus of this training is on point surveys. Collection method. So I said I did different here. So we'll say I captured or at least tried to capture only bees that looked different. Your area should be approximately 1 hectare. Number of surveyors, just me. I started at 12:45 p.m. I ended at 1:45, up to 1:45 p.m. Survey length in minutes. It should be a 45-minute survey. These times are further apart than 45 minutes. They're an hour, but I'm accounting for stopping my timer to transfer bees from the net into the vials and then starting again. So that took me, we'll say, 15 minutes. My temperature, 68 degrees. Fahrenheit. Cloud cover was, I estimated, about 15%. Wind speed said 5 miles an hour. I think it was probably 0 to 5 range. Weather notes, I don't have anything to say in there, but if you did, you could write something. Habitat type of survey area. So I'm looking over here. I have grassland and meadow. Primary habitat type of surrounding area was, let's see, I wrote grassland, was grassland and meadow. Optional secondary was shrub scrub. And then I didn't write a tertiary. There were really only 2, I'm going to say, in the survey. How much of your plot that you actively searched had flowers? So I said 60%. Then we'll go down here to nesting resources. I said there was bunch grass. That's not bunch grass. Bunch grasses, evidence of rodent holes and tunnels down here, and then pine needle and duff layer. Next, I'll go to the management activities section. Evidence of mowing. Said no. Livestock grazing. I saw footprints. If I saw others, I would check multiple. None of the above is also an option. Evidence of insecticides. No. Herbicides. Nope. Evidence of fire. We're saying no. How many honeybees did you observe in 10 minutes? I'm going to say I saw 12. Management notes, here you could write something like, if you said, I suspected I saw fire, evidence of fire, you could write in notes here why you suspected that. Number of flower species, I had 3. I'm going to go to the second page of my data sheet here. Plant 1. Look up potentilla, and it snazzy new feature should populate. There we go, potentilla chrysalis. It will have the little photo there. And then my second was lupinus. So I'm just going to say genus. I didn't know what species it was, so we're just saying it was lupin. Percent cover. Oh, and I have one more here. Pedicularis. There we go. I selected it there. Then percentages. I'm just copying this over from the top part. So 60%, 25%, and 15%. We'll say yes, the survey was a volunteer effort. So now I'm entering this information from the bottom. What time did I start? I started at 10:30 a.m. I ended at 4:15 p.m. How many miles did I drive? I drove 65 miles round trip, and I spent 1 hour organizing and entering my data, which is what I'm doing right now. Did I use online resources for training? We'll say, sure. I watched a webinar. On what day did I do that? We'll say I did that this morning. How many hours did I spend watching the webinar or reading materials? We'll say an hour and a half. So this next question, is this a null survey? So did you survey for bees but didn't actually detect or capture any? That's still really important information for us to know. So you should still be entering a survey even if you didn't find any bees. But no, this was not a null survey. I did find bees. Typing my name below. All information provided is correct and accurate. So now this is going to take me to this checklist. So I've now created a checklist, which is basically the survey. And now I can enter my sightings of my bees. So I'm going to go over here. I have this folder with photos of my bees. I also, on this data sheet, you can see the photo numbers associated with each of my 2 species that I saw, the 2 individuals. So we'll go ahead and select species or select photos. So here we are, and these top 4 are a single species. I'm going to select all of those. Those photo numbers should line up with these from the top row. We'll say open. They should all load. I'm saying in this data sheet that I didn't know what species this was. So I'm going to say bumblebee species. Someone else will identify it. Something I can do is use this identification tool. So this is a guide that will help me think through what species it might be. And this is helpful on our end for identifying bees to have a sense of what someone thinks it might be, getting it kind of in the neighborhood. If you select one and are not completely sure, that's fine. This is going to be identified by an expert. And so it's not going to end up incorrectly identified if you didn't select the right species here. But here we could fill in different information, doing this at random, and that will narrow down. You've seen whatever I selected, there are no matches. You can select these different body parts, and then it will give you possible matches. And then you can select the species and it will populate there. I'm going to say cancel. I'm leaving it as bumblebee species. I counted 1 reference ID. That's if there's an ID associated with it from some other project or something. I'm going to leave that blank. Sex, I know from looking at the characteristics of this that this is male. If you don't know, you could say not determined. And your options are queen worker, male, female, or not determined. And again, we will be checking this information, so it's okay if it's not correct. Floral host was Rotundilla Chrysalis under cinquefoil. I don't really have any notes there, so I'm going to leave that blank. Observational notes, I don't have any notes there either. If this was Western or Morrison's bumblebee, this is where you would enter the timed first detection in this area. We're going to leave this as public. That means everybody can see it. They should be left to public. We'll say add sighting. It will pop up here saying this is my first bee. Then we'll go ahead and go here. And I'm going to select my second bee. I had 3 photos of this one. This one I know is a Boswellsii. So I'm going to go ahead and select that. Count is 1. This looks like a worker with a lot of pollen on its legs. Floral host was a lupin, not sure, just genus lupinus, so we'll enter it there. I don't have any other notes, so we'll say add sighting. So there we go.
Can you just pause it for 1 second?
Information associated with the survey.
I just wanted to let folks know, Molly showed you the private option there a little bit ago, and most of you will be submitting your data as public, as you should, which is great. If you are doing surveys in Southern Oregon within the range of Franklin's bumblebee, Bombus franklini, and you think that you may have captured a Franklin's bumblebee, we have an agreement with the Fish and Wildlife Service that we're going to keep those sightings private until they've been fully documented. We don't want someone to find the location of that bee and then go love it to death because it hasn't been seen since 2006. So if you think you've found a Franklin's bumblebee and you're within the range, which is basically Mount Ashland down to the California border in Oregon, or I'm sorry, Roseburg, basically down to the California border. in Oregon, in that southwestern corner of the state. Please mark it as private. And if you would also reach out to us and let us know that you think you found it. We also have an agreement with the Fish and Wildlife Service that we will not publicize this. So please don't share any such finding on social media. There will be a time and place where we will share that information, but please don't rush out ahead of it. We just don't want to love that bee to death should we redetect it. Thanks, Molly. I just wanted to get that information in while we were on that part of the presentation. Thanks.
Uploaded into this checklist, and I have my 2 bees here. If you need to edit information related to a bee, you could click edit. You can also delete if you accidentally deleted the wrong, or you accidentally entered the wrong species and want to change that. You can do it here. I also just wanted to show you now where that would show up. So here, I went back to the home page and went to Bumblebee Maps. This brought me to the master sheet, or the master map showing bumblebees all years, all places. And my bumblebee is not, I'm going to try to find it. It's not verified. So we're going to say it's pending. So it hasn't been reviewed yet. It is in Oregon. It's collected in 2025. Let's see what that narrows me down to. Oh, not very many. I could also enter observer here. So I can type in my name. That narrows it down even more. Okay, so here are my bees. I can go and click on it, and it will show me these are my 2 bees that I've entered. So anybody can come look at this. It shows you they're pending, not verified yet. I can go to view sighting, and it will open up the sighting with all the information for that bee. You can also view the checklist, and that will take you to this survey, and you can see information about the survey and your bees. And you'll be able to see when they do get identified, that pending will change to verified or tentative. Likely verified, but it may be tentative if we couldn't see all the features necessary to identify it. All right, I'm going to transition back to our slides. Okay, I'm going to pause us there. That was pretty much everything from that. I don't know how to, hang on. Here we go. Okay, so that was basically all the content we have. This is just a reminder that on our website, we have all the recorded segments available, so you can go rewatch any of these that you want to revisit. We also have the participant handbook online, which has kind of a distilled version of everything you might need to know to be part of the Atlas. And then just a reminder, if you get off of this call and are excited to adopt a site, the first step you should do is go to the Bumblebee Atlas website, adopt a grid cell, look at this map, and then go ahead and pick which site you want and adopt it. And then the last thing is that you can connect with us and share your experience on a variety of platforms. So we are on Facebook, Instagram, and Blue Sky. And you can see here what those links are and how to find us. And then with that, I think we have 5 more minutes. So if anyone has questions, it looks like we have a few open ones here in the chat. If you have questions you haven't put in yet, put them in Q&A. Looks like Rich, you had one you were going to answer. And I saw one earlier, I think it disappeared, but somebody asked if they do surveys outside of our official windows, like for example, before June 1st, because a lot of areas that dry out later have flowers before June 1st, you're very welcome to enter those online. having data from any time of year is helpful. So go ahead and enter that and we'll use it. And then there's one other question. I think someone said that this project sounds great, but it's a lot of work to do all of the surveys throughout the year. But if you want to continue just doing surveys wherever you like and not in Sentinel sites, that's still great. And we'd like that data. Rich, did you want to answer this one you had flagged?
Yeah. Someone just asked a question here. Is it acceptable to change survey like single hectare sites within the adopted grid cell from one time period to another? So in other words, we have these large hexagonal grids. Ideally, you would pick a 1 hectare site within that, 7.5 kilometer area, and you'd survey that in each of the 4 time windows. So ideally, you'd pick a site that would be sufficient for those 4 time periods. That isn't possible in all regions of our 3-state area where stuff is done by June in some locations, right? Particularly certain years. So, the ideal is for you to pick one site and survey it all 4 windows. If you show up at your site and it is just a dried out mess and there's nothing there, and you know that you're not going to find bumblebees, but there are other survey locations nearby that do have flowering resources and bumblebees, I would say that as long as you stay within your hexagonal grid, that you can move sites. Ideally then, let's say you go for your 3rd site, and then you move in that 3rd window. In the 4th window, you'd go back to that 2nd site that you used. So try to repeat as many times as you can in as many places as you can. I will also say that if you know that your site is going to dry out, like you had this experience last year, the survey windows are sort of general guides, and this is probably not something I should say out loud to a large group of people, but if you know that your site's going to dry out and you want to do 2 surveys within window 1, because that's your opportunity, that's the window for that site, then please do it. Those data are great, and we would love it. What we're trying to do is get them spread out throughout the season in a large 3-state area, and that's hard. It's hard to make recommendations across that area, and you all know your local sites really, really well. So we've provided this general framework, but ultimately what we want is as many repeat surveys in the same location during the peak of the survey season as we can possibly get. And I realized I just said that really fast, but hopefully you understand what I'm saying. And so there is some flexibility in there within the larger framework. Like if you get it in like a day early or a day late within a window, like it's not going to break the system or anything. Like it's a framework, follow it to the best of your ability. And if you need to break that, break it. And if you have specific questions or want to follow up, please reach out to us. During the field season, it's best to just email pnwbumblebeeatlas@xerces.org, because Molly and I have other projects going on. We're in and out of the office. That one goes to both of us, and it's more likely that one of us will be able to respond to you. So yeah, I hope that helps.
Yeah, I think that's a great point. And probably a good question to end on. It looks like time's up and we don't have any other open questions in the Q&A. So thank you everybody for being here tonight. And please reach out to us, like Rich said, email us anytime if you have questions, and we're happy to help.